Transplantation and tumor immunology. Transplantation. Transplantation is a useful process for replacement of a diseased tissue or organs which are irreparable damage. The tissue or organ transplant is called transplant or graft. Types of transplant: autograft. It is a tissue or organs taken from one side and engrafted to another side in the same individual. Isograft. It is the graft taken from an individual and placed on another individual of the same genetic constituent. Example: graft made between identical twins. Allograft. It is a graft transfer between two genetically non-identical members of same species. Xenograft. These are grafts between members of different species and are called xenograft. Autograft and isograft are usually accepted and survive. Allograft and xenograft are usually rejected due to the genetic and antigenic incompatibility. The allograft reactions. The rejection of the graft by the recipient is called the allograft reactions. Histocompatibility antigen. The histocompatibility antigen or specific for each individuals and or under genetic controls. The term major histocompatibility complex is a system of cell antigens that are responsible for allograft rejections or acceptance. The MHC in man is a human leukocyte antigen system. Graft versus host reactions. The graft rejection is generally due to the reactions of host to the grafted tissue. Host versus graft response. The contrary to that, the graft may mount an immune response against the antigen of the host. This is known as the graft versus host reaction. The graft versus host reactions occurs when the following conditions are present. The graft contains immunocompetent T lymphocytes. The MHC antigen of the recipient activate transplanted immunocompetent T lymphocyte which leads to the production of lymphokines. The T cells antibody. They attack the recipient cells, leading to manifestations of graft versus host reactions. The recipient possesses a HLA antigen that are absent in the graft. The recipient immunological the responsiveness is either destroyed or so impaired following whole body irritations that he cannot reject a graft. The graft versus host reactions are predominantly cell mediated. The manifestations of graft versus host reactions consist of splenomegaly, fever, rash, anemia, weight loss, and sometimes death. The neonatally thymectomized animal receiving an allograft of spleen or blood lymphocytes do not grow normally but develop a fatal wasting syndrome known as rusting disease. The tumor immunity. When a cell undergoes a malignant transformation, it expresses a new surface antigen and may also lose some normal antigen. The tumor-associated antigens are immunologically distinct from normal tissue antigens. The host recognizes the tumor-associated antigens as non-self. Therefore, the tumor can be considered as an allograft and it is expected to induce an immune response. Tumor antigen. A tumor-specific antigen. The tumor-specific antigen are present on the member of the cancer cells and not on the normal cells. Such tumor-specific antigens reject tumor transplant in immunized host. These are referred as a tumor-specific transplantation antigen or tumor-associated transplantation antigen. The tumor-associated antigen. These are present on the tumor cells and also on some normal cells. These includes the tumor-associated carbohydrate antigen. They represent the abnormal form of glycoprotein and glycolipids such as a mucin associated antigen detected in the pancreatic and breast cancer. Oncofetal antigens. These are fetal antigens which are present in the embryonic and malignant cells but not in normal adult cells. The best known examples are alpha fetoprotein in hepatoma and carcinoembryonic antigen in colonic cancer. The differentiation antigens. An antigen expressed in early B lymphocyte is present in B cells leukemia. Similarly, the prostate specific antigen has been used as a diagnostic a indicator in prostatic cancer, immune response in malignancy. The delayed higher type hypersensitivity T cells plays a significant role in tumor killing by means of lymphokines that they releases. The tumor cells can kill the tumor cells in the absence of antibody. Natural killer cells also participate in the antibody dependent cytotoxicity. The humoral response may also participate in the tumor cell killing, the activations of complementation and induction of ADCC by natural killer cells. The immunosurveillance. It is believed that the malignant cells arises by mutations of somatic cells that occurs frequently. It is postulated that the immune system keeps a constant vigilance on these malignant mutations of the somatic cells and destroy them on the spot. The inefficiency of immunosurveillance, either as a result of aging or in congenital or heterogenic immunodeficiency, leads to an increased incidence of cancer. The development of tumors represents an escape from this surveillance. The mechanism of such escapes are not clear, but various possibilities have been suggested. This includes the modulations of surface antigen, masking tumor cells, productions of blocking antibodies, fast rate of proliferation of malignant cells, suppressions of cell-mediated immunity.